Welcome back. In this web lecture, I'm going to briefly introduce you to the structure and development of the most fundamental elements of the axial skeleton, the vertebrae and ribs. So as I mentioned in class, the very earliest function of the vertebrae was to cover and protect the neural tube. The very earliest structures that we see evolutionarily in terms of vertebral elements is this neural arch that's going to grow up and over and cover the neural tube, giving it some protection. So these neural arches often bear a structure called a neural spine at the top, a little projection off of the top that generally functions as an anchoring point for muscle, so for muscle attachment. So a little bit later in evolutionary history, we also start to see ventral vertebral elements. And these are going to grow down ventrally and surround the dorsal aorta, and these are known as hemal arches. So often these will also have projections on them known as hemal spines. So these are the very, very earliest functions of the vertebrae. At this point, the notochord still has the primary responsibility for maintaining the body axis, maintaining the body length, making sure that the body doesn't shorten when those body muscles contract. So later on in evolutionary history, we start to see a more complete vertebra. So we start to see the appearance of a solid bony structure joining these hemal arches and neural arches called the centrum. And so as you saw in your book, and as we'll discuss a little bit further in class, the centrum can take a number of shapes that reflect its functional properties. And here you see in the fish vertebrae this hemal arch with a hemal spine in the tail vertebrae of the fish, and also projections coming from this centrum known as transverse processes, also functioning in muscle attachment. In the cartilaginous skeletons of sharks, we see a very similar structure, just composed of cartilage. So in sharks, we see again the vertebral arch that forms this kind of tube-like structure over the top of the centrum. We see a hemal arch ventrally, and then in sharks we start to see much more complete coverage and encasing of the nerve tube. So there are also these cartilage structures called neural plates that are going to come up and between these neural arches, and then also between sequential neural arches, structures that we call intercalary plates that are going to cover over the top of what otherwise would be the exposed portions of the nerve tube. So in sharks we see this very complete coverage and encasement and full protection of the neural tube. So the spinal cord is going to be coming through this canal right here. This is a solid cartilage centrum that is going to now provide that structural axial support rather than the notochord doing that. And then again, the hemal arch is going to be protection for the dorsal aorta. At this point, it's the caudal artery. This is an example of a tail vertebra. And then also the caudal veins are going to travel through these hemal arches in the case of sharks. So with this complete coverage and this complete encasement of the spinal cord, clearly there needs to be some way for the spinal nerves to exit and spread out to get to their targets in the body. So what we see in these neural plates are these little perforations or these holes, also called foramina. Foramen translates as window. So it's a little hole or window in the neural plate for the spinal nerves to exit. So we also see in amniotes greater protection for the neural tube than we saw previously in bony fishes. So these neural arches can be greatly expanded, and when adjacent neural arches come together, they fit very closely with just a hole in between them, and this is known as an intervertebral foramen. So these are separate vertebrae that articulate together, but they come in very close association, just leaving this hole for the exit of those spinal nerves to come through. Otherwise, the neural tube is completely protected by these neural arches that are widened and expanded. So these neural arches have these interlocking processes. So hopefully you saw this in lab, called zygapophyses. So each vertebra has a cranial zygapophysis on the anterior end that's going to articulate with a caudal zygapophysis on the adjacent vertebra. So this is the caudal zygapophysis for the more anterior vertebrae, articulating with the cranial zygapophysis. So this caudal zygapophysis will articulate with the cranial zygapophysis of the next vertebra in line. So now let's start to think about how these structures develop. So we know that during the differentiation of mesoderm from this paraxial mesoderm along the sides of the neural tube and notochord, we get condensation of these little balls of tissue into somites. This is the basis for segmentation. So vertebrae and ribs are clearly segmental structures. They're going to come from these somites, and in particular they're going to come from the portion of the somite known as the sclerotome. It's going to give rise to both the vertebrae and the ribs. So let's take a look at how this happens. So very early in development, some of the first structures that start to form to organize the trunk of the body are these membranous septa. So we call these skeletogenous septa in various places in the body. So you saw some of this in your lamprey and also in your shark. So you would have seen in your lamprey this dorsal septum that separates the right and left halves of the body. In your shark, you saw the horizontal septum that separates the apaxial muscles dorsally from the hypaxial muscles ventrally. So this is going to run all along the length of the body. And then there are these myosepta that separate the different segments of segmental body muscles. So the somites are going to form kind of in between these myosepta, and the muscles are going to fill in these spaces in between the myosepta. The myosepta are actually the tendons that these muscles are going to attach to in the body wall. And there's also a lateral septum that's going to surround the coelom and the gut tube. So what these septa are going to do is they're going to provide pathways. They're going to be sort of the highways along which these mesenchymal, or in other words, migrating cells, are going to travel to find the exact spot where they should condense and start to form these skeletal structures. So let's see how that works. So in this first part of the figure, we see this somite that's differentiating into dermatome laterally, myotome in the middle, and sclerotome medially closest to the neural tube and notochord. And so if we take a look at these segmental somites, each one of these represents a ball of tissue. They're color-coded so that you can keep track of the origin in the somite of these particular cells. Because what's going to happen as these cells become mesenchymal and start traveling to their destinations is that they are going to split up and undergo a process that we call resegmentation. So they're going to maintain their segmental identities, but they're going to join up with some of the sclerotome cells from adjacent somites to form a newly segmented structure. The sclerotome that retains its original segmentation, so this pink one, this blue one, and then this pink one, each of these is called primary sclerotome. This is before this process of resegmentation. And then the resegmented sclerotome is known as secondary sclerotome. And so this is what's going to come and start to surround the notochord. Each one of these secondary sclerotome structures is going to be made up of the posterior part of one of these somites. So this is the pink that's going to come and form this part of the secondary sclerotome, and the anterior part of the adjacent one. So some from this somite, some from that somite are going to come together to form the secondary sclerotome. So the sclerotome is going to start to form these ring structures around the notochord called paracortal rings. So you see that right here. These paracortal rings are going to grow and expand until they form a paracortal tube that's going to completely surround the notochord. Shown here in the dashed lines are the positions of those originally segmented areas. So there's the location of one of these primary somites. There's the location of the next one. And as these vertebrae form, each vertebra is going to be formed in this resegmented way, half of it coming from one somite, the other one coming from the other half. And in the middle of each of these, we're going to have the formation of these intervertebral discs. So the vertebrae are actually what's known as intersegmental structures. They're off register with the rest of the segments. And the reason for that is these muscles that form are going to attach to the vertebrae. So these are representing the axial muscles that are in line with each of these original somites. And so if they were in register with the vertebrae, they would be attaching both ends of the muscle to the same vertebra. They couldn't cause any movement that way. For muscles to create movement, they have to cross a joint. So in this way, with this resegmentation of the vertebrae, these axial muscles are going to attach to adjacent vertebrae, one end to each of them. They're going to overlap the vertebrae and actually be able to cause them to move at this articulation between them. Now let's take a look at how the ribs form. So we mentioned all of these different septa. So what's going to happen is we're going to have sclerotome traveling out along these little passageways, these highways formed by these septa. So remember, in each one of these compartments, this is the location of the myotome. We're seeing it without the muscle filling this in, but you can imagine a segmental muscle here, segmental muscles here in the hypaxial region. This sclerotome, mesenchyme, is going to gather at the intersection of this horizontal septum and each of the myosepta. So these are called intermuscular ribs or dorsal ribs. Ribs are also going to form when sclerotome migrates along each of these myosepta adjacent to this lateral septum surrounding the coelom. And so these are known as ventral ribs or subperitoneal ribs. And so many fish have both of these pairs of ribs. So if we look at the very simplified axial skeleton of a fish, what we see are trunk vertebrae. So these are going to have transverse processes that are going to articulate with both these intermuscular ribs. You can see the dorsal ribs kind of sticking out here, and the ventral ribs or the subperitoneal ribs coming down and surrounding the gut tube. And these transverse processes are going to be the articulation point between the vertebrae and the ribs. And then they have tail vertebrae. And so rather than the transverse processes, they have hemoarches in the tail. So you'll see hemoarches and spines instead of transverse processes. So these transverse processes and hemoarches are what we call serially homologous. They are homologous structures. They just take different forms depending on whether they're in the trunk or in the tail. And so the reason that we maintain these hemoarches in the tail of fish is that fish use their tails to vigorously whip back and forth during locomotion, and that's a part of the body where they really need to protect that blood supply to the tail because of the high activity level in this region. In the trunk region, the dorsal aorta is already very well protected by the internal organs. It's surrounded by soft tissue. Doesn't need to worry about it being crushed quite so much. But in the tail, we use those hemoarches to protect that blood supply. So we'll see in class, in tetrapods and other vertebrates, these vertebrae are going to become much more regionalized, much more differentiated, and take on many other functions other than just articulating back and forth the way we see in a fish.